Every living thing on this planet, whether it's a tiny bacterium or a massive blue whale, is built from this incredible instruction manual called DNA. It really is the code of life itself. But here's a wild thought. What if we could actually edit that code? It sounds like something straight out of science fiction, right? But believe it or not, scientists have been doing exactly that for decades. And to pull it off, you can't just, grab any old pair of scissors and glue. Nope, you need something much, much smaller. What you really need is a super specialized set of biological tools. We're talking about a molecular toolkit, one that's packed with amazing little enzymes that can cut, copy, and paste DNA with just incredible precision. So in this explainer, we're going to open up that toolkit and take a look at what's inside. So our toolkit is organized into 4 main trays. First up, we've got our cutters. These are the nucleases. Then you have the ligases, which act as our molecular glue. Next are the polymerases, which are basically our copy machines. And finally, you've got the modifying enzymes that handle all the important prep and finishing touches. Let's kick things off with the cutters. Okay, so the very first step in pretty much any kind of genetic engineering, and this is absolutely crucial, is making a precise cut. You can't just chop up DNA randomly. You need molecular scissors that cut at the exact spot you want. And these amazing tools are called restriction enzymes. What you're seeing here are the 2 basic ways to get the job done. Over on the left, you've got what we call exonucleases. They kind of just nibble away at the ends of a DNA strand. But the real stars of our show, the ones we really care about in our toolkit, are on the right, the endonucleases. These are the ones that can make a clean cut right in the middle of the DNA molecule. So where did these incredible tools even come from? Well, it turns out nature beat us to it by a long shot. Bacteria actually evolved these enzymes for one very specific reason, as a defense system. They use them to chop up the DNA of invading viruses, like these bacteriophages you see here, before they can take over the cell. It's basically an ancient form of biological warfare. So the big question is, how do these enzymes know where to cut? They don't have eyes, right? Well, what they do is recognize a very specific short sequence of DNA letters. And these sequences have this really cool property. They're palindromic. Like the word race car. The DNA sequence GAATC on one strand is matched by CTTAG on the other, which if you read it backwards is GAATC. That's the signal they're looking for. And here's how it works. The enzyme shape is tailor-made to perfectly lock onto its unique palindromic sequence. Once it finds that exact sequence, and only that sequence, it latches onto the DNA, ready to make its cut. It's this incredible specificity that makes the whole field of genetic engineering even possible. All right, but here's a twist. Not all cuts are created equal. An enzyme can slice the DNA in 2 different ways, and this produces either what we call sticky ends or blunt ends. Now, this might sound like a tiny detail, but it is so important for what comes next because it completely changes how easily we can build our new DNA molecule. Let's take a closer look. On the left, you can see how making a staggered cut creates these single-stranded overhangs. We call them sticky because they're just itching to pair up with a complementary sequence. Think of it like molecular Velcro. Then, on the right, you see a straight cut, which creates a blunt end with no overhang at all. And as you're about to see, those sticky ends are the real key to easily slotting a new piece of DNA into the gap. And just to make things more interesting, not all of these restriction enzymes are built the same. Some are, well, a little sloppy. They cut randomly, pretty far away from where they bind. But the ones we love, the real workhorses of genetic engineering, are the type 2 enzymes. And here's why. They cut with surgical precision, right within their recognition site. They're the reliable, dependable tools that make all this magic happen. All right, so we've made our perfect, precise cut. Now what? We need to paste a new piece of DNA into that opening. And for that, we need to pull another tool out of our kit. Our genetic glue, a fantastic little enzyme known as DNA ligase. So in a cell, DNA ligase is kind of like the on-call repair crew for the DNA backbone. Its job is to fix any breaks or nicks it finds. In the lab, we totally take advantage of this natural repair function. We use it to permanently seal our new DNA fragment right into place, creating one single, continuous, and totally stable molecule. The process itself is actually pretty straightforward. It's just 3 steps. First, the enzyme zips along until it finds the break. Second, it uses a little bit of chemical energy to prep the site for repair. And third, it forges a brand new, super strong bond, making the DNA whole again. Simple as that. Okay, I know this diagram looks a little complicated, but don't worry, it's really just showing that simple three-step process we just talked about. The ligase finds the gap, it uses an energy molecule, ATP in this case, to get a grip, and then it uses that energy to create the final permanent bond. It's truly the molecular equivalent of a perfect weld. So, we've cut and we've pasted. We are on a roll. But what happens if you only have one tiny little piece of the DNA you're interested in? You're going to need way more than that to work with. You need a way to make millions, even billions of copies. And for that, we turn to the third set of tools in our kit, the molecular copiers, aka DNA polymerases. A DNA polymerase is literally nature's own copy machine. Its whole job, its entire purpose, is to read an existing strand of DNA, which it uses as a template, and then build a brand new, perfectly complementary strand right alongside it, one building block at a time. But there's a catch. A polymerase can't just start copying from anywhere it wants. It needs two things to get going. First, the template strand to read from, and second, a short little piece of DNA that's already attached, called a primer. That primer gives the polymerase the starting line it needs to begin building the new strand. Now, scientists have a whole bunch of different polymerases in the toolkit, but there's one that was a total game changer, Taq polymerase. This one comes from a bacterium that lives in scorching hot springs, so it can handle incredibly high temperatures without breaking down. And that special heat resistance is the secret ingredient that makes a powerful DNA copying technique called a PCR possible. It lets us go from a single copy of a gene to millions of them in just a couple of hours. It's amazing! All right, we've seen our cutters, our gluers, and our copiers. We've looked at all the individual tools. Now for the really cool part. Let's see how they all come together to build something completely new. Before we put everything together, there's one last little tool we need to talk about, the modifying enzymes. Think of these as the finishing tools, the sandpaper and primer you use before you paint. For example, an enzyme called alkaline phosphatase does this really clever trick. It basically snips a little chemical group off the end of our DNA vector, which prevents the vector from accidentally gluing itself shut before we get a chance to pop our new gene in. It's a small step that makes a huge difference. And here it is. This is the payoff moment. We take our restriction enzyme, our scissors, and we use it to cut open this circular piece of DNA called a vector. Then we use the exact same enzyme to cut out the gene we're interested in. And because they were both cut with the same tool, they now have compatible sticky ends. They just slot together perfectly. And then, bam, DNA ligase, our glue, comes in and seals the deal. And the final result of all this cutting and pasting is a brand new hybrid molecule we call recombinant DNA. It's a custom-built piece of genetic code that literally combines DNA from different sources, and it's ready to be put into a cell to do something new, like programming bacteria to produce life-saving insulin or giving a crop the power to fight off a disease. So from bacterial defense systems to enzymes found in hot springs, this amazing molecular toolkit gives us a power that's, well, it's unprecedented. It's the ability to directly engineer the code of life itself. And that leaves us with one really big question, doesn't it? What do we choose to build with it?